So I'll start the recording. The whole idea of this activity is to kind of get used to these cladograms and constructing them, looking at the differences in phenotypes between different organisms and how evolution might have played a role in distinguishing these species. So as you can see, we're looking at corn, some type of fish, it looks like, platypus. Those are really interesting animals, honestly. Um, and then penguins, and then it looks like some sort of, yeah, HIV and then E. coli. Okay, I see. So it's salmon for the fish. So down here, you're looking at different characteristics. As you can see, there's a list here that you'll notice has some differences between all of the different species or viruses. And you'll also notice that some of them are eukaryotes and some are prokaryotes. And you'll see that all of these pluses are a phenotype that the specific species has. So here you'll see that corn has cells, or like it's a cellular organism. And not only that, but it's a eukaryote because it has a nucleus. And then also, its genetic material is made up of nucleic acids. So you'll look at all of the differences between these organisms. And then look in question one, it asks based on the table above, complete the number of differences between each pair. So here you have a couple of different examples. Comparing A and B here in this top left box, you'll see that's comparing corn and salmon. Now you'll see that both of these individual species do not have feathers, hair, or gizzards. So there's no differences there. Also, they do have cells that have a nucleus and that their genetic material is made up of nucleic acids. So there's no differences there as well. And both also do not produce amniotic eggs. But one difference is that a salmon has a jaw, while corn do not. I mean, that'd be kind of scary if like corn had jaws. I don't know how I feel about that. But there is one difference here, so that's why you have one on the example there. And then same thing, comparing E and F, which is E. coli and HIV. You'll see that E. coli has this extra plus for cellular, while HIV does not. But all of the rest is the same. Either they do or don't have pluses, which gives that one difference. Now you're going to compare all of these different organisms to one another, and you only have to fill out these white boxes. A lot of students usually fill out the darker boxes as well, but that table or that half of the table is pretty much like blacked out because it's just going to be a mirror image of what you find up here. So that's just unnecessary work. For example, you'll be comparing B and B here, which doesn't make sense. You're going to have zero differences, and that's not needed. And then also, let's look at this box too. You see that you're comparing C and B. That this number is just going to be here. So like I said, it's just going to be a reflection there. So yeah, once again, don't fill out, or you don't need to fill out the dark colored boxes. Now here, in question two, circles indicate where the species from the table should be located. So you'll see that uh, each of these circles should have the letter of the species. So you'll have A through F somewhere up here. And then dotted lines indicate missing characteristics or features. A lot of students forget this part, but you want to make sure that you put characteristics here on this dotted line. So, for example, one of these is cellular. And so you'll notice that the difference between all of these five organisms and this one is that this one is not cellular while the rest of them are. So you should be able to get that from the table. And then you'll just make those, um, you'll just distinguish the differences between like all of these four species on the left and then these two species on the right with this little dotted line here. And then you'll also have to use these one, two, three, and four boxes for question three, which is match the following components of a rooted tree with the respective numbers in the squares from the tree on the right. So you'll look at, there's four vocabulary terms here, internal nodes, root, terminal nodes, and branches. And using these numbered boxes here, you'll kind of like denote what that box is pointing to. So for example, three is pointing to this line here, which is one of these four. Whereas the shelter, shelter. Sorry, I might be misunderstanding your question, but what does shelter mean?
Sorry, never mind.
No.
I was asking Jeanette a question.
Oh, you're good. You're good. You're good.
Responding.
Oh, the dog shelter. I was like,
Yeah.
man, shelter. I'm definitely missing something. No, no, no. You're good. Uh, yeah, where is the shelter, Jeanette? I'm just kidding.
It's in Northwest Indiana,
Oh,
so I'm
awesome.
about an hour away from Chicago.
Oh,
Wow.
that's really cool. Lakshmi, did you have a question? I saw you unmuted for a little bit.
Yeah, so just wanted to, you know, do we cross-check our answers with you, or how does this work
So the answers, you
for the
can.
table,
Sorry, what was that?
for the table on the left?
For the table on the left, cross-check. Do you mean just like checking with one of us before you turn it in?
Yeah, so just for the first row, I mean, can we, because there was a first version and it had mistakes, then I downloaded the second version of this. So I got a little bit wary. I don't know. I just wanted to make sure. So A to C, the difference is three, right? Is that how you want to look at it?
Yes, that's how you want to look at it.
Yeah, okay.
I believe the first version just had like one extra plus. That's
Plus
at
for
least what I
F.
saw.
Yeah,
Yeah, yeah. Um, so the entire table should be the same except the comparisons with F
F. Okay.
and yeah, some of the species.
Okay, so another question. For this, for an exam, right?
Mhm.
If they give the top table, I think I can form the cladogram. But if it's on the bottom left, right, this table, I don't think that helps me well to answer these questions.
Right.
I
I
don't know why.
mean, so the only thing I can think of what you can do is find like the like distinguish the number of differences between certain organisms, and you'll know that the more differences there are between like one organism versus the next means that they're going to be on like opposite sides of this tree.
Oh,
Um,
awesome. Okay.
I don't think ATAs, you can correct me if I'm wrong, but I don't think there's any questions going from like a table like this with the number of differences to actually constructing some sort of tree.
Not that I remember.
Okay, perfect. That's a good question though, because that would be a tough one on an exam. So
Yeah, thank you.
yeah, yeah, of course. In terms of checking answers on activities, I mean, you can email the us TAs or maybe even the ATAs, but we can't actually like give you the answers. We can just kind of like confirm if what you're doing is like on the right track or like if your thinking is pretty sound, you know?
Awesome.
Yeah. Does anyone else have any other questions about activity two? Well, if you all feel good about activity two, you can ask other questions as well about the class. I'd be happy to answer those. Or if you're like actively working on activity two, you can ask questions if anything comes up.
So, look, I have a question on Mendel's laws, the concept quizzes, on questions. I don't know if they are jumbled up from your end, but it's problem 5 and 6. I mean, so that's where I have some doubts.
Gotcha. So you said quizzes 5 and 6.
5 and 6 for Mendel's law.
Okay. And that's on module 2 as well.
Yeah, module 2.
Perfect. Yeah, I'll go over there and then I'll answer the questions in the chat after this. Say 5 and 6. Okay, so looking at this concept quiz here.
Yes, on
Okay.
Mendel's laws.
Mendel's laws. Let's see here. So is there, which
No,
question
no,
is that?
not this. Mendel's laws. This is
Mendel's
yeah,
laws.
yeah.
Okay, I see. Sorry. I
No,
don't
no, no
spend too much
problem.
time with the concept quizzes, so I'm a little lost. Gotcha. Okay, so this one right here.
Yeah.
Okay, perfect. Okay. So which question
5
specifically?
and 6.
Okay, 5 and 6. Okay. So how do you feel about the idea of like what the gamete genotype should look like from an like
Yeah. So according to me, uh, so since there are two of the same dominant A, right?
mhm.
So that's one gamete, if I'm
O-
right.
Okay. Right. So like all of their gametes will have big A.
A. And
Mhm.
so the uppercase B and the lowercase b would be two separate. So there'll be two,
Okay.
and uppercase C and lowercase c will be two. So total 5.
Okay. Let me see what you're looking at. I think that's a pretty good way to think about it. Um, yeah, so like you said, every gamete is going to have big A. So then you're just looking at, right, like all of the different possibilities of big A and then either big B little b or big C little c.
Right.
Yeah.
So the combination would be A, uppercase B, C, like that. And A will be constant for all of them.
Yes, correct. Yeah.
Okay.
Yeah, so if you write all of those out, you should be able to get
Okay.
that answer. Yeah.
And I'm a little bit lost with question 6.
Question 6. Yeah, sometimes with these like long questions, it's hard to follow along. Okay. So orange is dominant to white, round is dominant to oval. To produce dihybrid offspring plants. Okay, so from this first part, when it says that two true breeding um plants, which it looks like plant A is homozygous dominant, right? For both or for both genes
Yeah, orange
and round.
and round.
Okay, perfect. So it's going to be all capitals and then or all uppercase and then plant B is homozygous recessive.
Yeah.
Just dihybrid offspring plant. Okay, so then the dihybrid offspring plant will be heterozygous for both genes.
Yes.
Perfect. So one of these two traits, which gametes might be produced by plant C? Okay. So plant C, is
So
going to have
heterozygous, right?
yes.
So yeah, so the gametes, so we got to that's where my question is. So I chose the first one, which is obviously not correct because I chose the actual genotype.
Yes,
Yes,
correct.
but I have to find the gametes for these.
Correct, correct. So there's looks like there's a few different gametes that are possible. Just looking at these answers, you'll see that this big O, big S, you see that that is a possible gamete, right?
Right.
And then same thing with this second one, because you have a little O here and a big S.
Mhm.
So it's possible during meiosis that plant C, when producing gametes, will,
Have
um,
both those.
yes, yeah, we'll give like a lowercase o or a recessive allele for locus O and then a dominant allele for locus S.
Okay, but my question is why are the other combinations are not listed, right? So that's where I got confused. So these are not the only
Gotcha.
gametes, right? There can be other combinations like uppercase O and lowercase s, and
Correct.
both or lowercase O and lowercase s. So why aren't those gametes listed? So that's where I got lost.
Definitely. I think it seems to me that um, I don't know why specifically not all of them were written. I'm not sure how Dr. Holacek normally writes, questions like
Uh-huh.
this, but
Uh-huh.
you just want to, like when you're looking at a question like this, even though it is a little confusing why certain gametes aren't listed, you just want to select the true answer regardless of what's available.
Okay. Okay.
Yeah,
Right.
yeah. So it wasn't like a trick question or anything where some of them were listed, some of them weren't.
Uh-huh. Okay.
Those, yeah, yeah. Good
All right.
question though. Yeah,
Right.
that was a really
Thank
good
you.
question. But yeah, you're thinking about like the gamete genotypes and everything like that is perfect. So
Thank you.
yeah, of course. Okay, so then looking at the questions in the chat, yeah, 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 Mary, the HIV does not have hair. So two. Okay, perfect. I'll respond to your email then, right after this office hour. Yeah, yeah, of course.
Hi, may I ask a question?
Yeah, of course.
For the extra credit, when signing up, what happened is we formed a group in the first week, but our group leader, she had to withdraw from the class. So she asked me if I could be the group leader. I
Okay.
was wondering if in um what's the difference? Would it be just be that the group leader has to turn in or submit the final um the final
Like the submission and everything. Like
yeah,
the okay,
that's
yeah.
the main difference?
Yeah, that's the only main difference. Basically, the group leader just turns in each draft submission. And then
Okay.
you would just organize with your group to make sure that you guys are on the same page. You would also go through like each section. So if you um assign like a group member to work on a specific portion for like draft one or draft two, you would just go through with all the other group members just to make sure it looks good before turning it in. That's it.
Okay. Also, I had another question. So before the class even opened, we started at, and I tried signing up for, on that website, and I couldn't for some reason. I know there were two options, so I tried the first one, I couldn't do it, and then I tried the second one, and I couldn't do it. But then I was reading through it. Does each member have to sign up, or I think I saw
So
somewhere
if
that
you're
maybe
having trouble
two?
signing up on all of us, reach out to us because we have an alternative training that we can assign you on Canvas. It's
Okay.
just let us know like what is causing the issue on the All of Us website. So email myself, Abby, Deidre, or Sarah, and we can assign you the alternative quiz when you give us like what's going on. Each member does have to at least do the alternative quiz or the training through All of Us for draft one.
Oh, three. Okay.
Yeah.
Okay, great. Sounds good. Thanks so much.
I have another question regarding the project with the data. I mean, obviously, there are a lot of restrictions that you cannot take a screenshot of the data and so on. So, but how do we proceed with what we are looking at from our, perspective with respect to our project?
There's
Uh,
different things. So like I have little videos that once the sign up ends on Wednesday, I send out the YouTube playlist that I have that shows like how to do each draft, how to sign up for all of us and what is needed and how you go from analyzing the data on all of us. And then Abby and I hold drop-in office hours each week. We just send out a notice of when we're going to hold them that go over each draft.
Awesome. Thank you. Thank you so
Mhm.
much. Just another question. So, is exam one going to be scary?
I think so session A, so literally just a couple weeks ago was the first time that I actually TA'd for this course. A lot of people were running into problems of like the time it was taking them to actually complete the entire exam. So you want to make sure that you work efficiently through all of the questions, even if there's some that you're not sure about, at least put an answer down and then if you have time, go back to it because the final question will be an essay question, and Dr. Holacek, at least when she goes over the exam, and the ATAs also will recommend that you do most of the essay question while you're still on part one of the exam. And then that will allow you to have all the information you need to actually work on it and submit part two of the exam within, I think, is it 30 minutes of the essay?
Yeah,
Um,
that's what is not
for the
clear. How, I mean, so you start the part two of the question while you're still on part one?
Yeah, the essay question is in the part one. Part two is where you turn in your scratch work and the short answers so the TAs can grade it for you.
Oh, okay.
And
yes.
then my best advice for taking the exam, if you can't answer that question within 10 seconds, like you can't think of how to answer it within the first 10 seconds, mark it and go to the next one and get the ones that you can actually do first so you get the points and then go back.
Okay. Okay. Thank you.
Mm-hmm.
Yes, definitely on the questions that require scratch work, please make sure on your submission for exam or the part two of the exam, the short answer part, that you do include all of your scratch work. Because if you don't and you get the question correct, we have to deduct half of the points, which, yeah, is unfortunate sometimes.
Will the exam review be similar, like format, like to understand better, like because I understand, but that is you have the question in part one. Will the so once the exam finishes, it will vanish, and is that what you're saying? So that we have it written down on our paper. Okay.
Yes,
Uh-huh.
yes. So it'll usually be like a pretty a somewhat complicated question where there will be a lot of information on part one itself about the question. And then you're right, as soon as the exam closes, you won't have access to it anymore.
Okay.
Yeah.
Thank
So like on your scratch sheet, people usually just jot down a bunch of notes, or redraw things that are on the question. Um, and then with that information there, or as well as the prompts to all of the specific questions. Like for example, um, one question can have like five other questions, like five different parts to the entire part two essay portion. So you want to make sure that you just have all that information down.
you. Okay.
Yeah. Just out of curiosity, Jonina, where are those exam practice questions? Are they on Canvas? And
Yeah,
they're
they're
just
in the exam module, so she'll
okay.
post them. She should post them. They should already be posted because I know she's going over.
Okay.
I think Wednesday is her exam review one. Yeah, I think Wednesday is the exam review. They should be in the exam module, so she'll
Hmm.
post a week before.
Sorry to ask you about this part as a part two of the exam again. So don't we have to show our answers to the camera before we close the the timing for the exam?
Yes, but as long as your short answer is about 70% done and you don't change majority of it when you turn it in, because the TAs will review your like video plus they'll review your answers to make sure you didn't change anything. So
Oh, okay.
70% completed and you just need to finish like a little bit. Like let's say you're on like part C of the short answer and time runs out and it you need to like show your answers. You can finish up part C as long as like nothing else changes.
Oh, okay. I get it.
Yeah. She's gonna go over it. She's gonna explain everything too in her one of her 3 office hours in the next 3 days.
Okay, no problem. The only thing is, I mean, because I'm going to be traveling, I might not be able to ask questions directly, and I don't know if the chat questions show up on the recordings. So that's what my doubt was.
They don't, but if you email her beforehand and have questions that you would like her to cover in the office hours, she will.
Okay, awesome. Thank you, Luke and Janina. I'll leave now. Bye.
Yeah, of course. Have a good one.
You too. Thank you. Bye.
Thank you. Hey Mary, I see that Aaron responded to your email. Does that explanation make sense?
I haven't actually checked it yet, not after I got on here, but let me check.
Okay. Yeah, that sounds
Okay.
good.
Hi, yes, I just saw it. I wasn't expecting that, but yes, that makes sense now that he explained it that way.
Okay, okay, perfect.
Thank you.
Yeah, yeah, I'm glad that helps. Um, yeah, distinguishing that difference between genotype and phenotype is just like, I don't know, it's kind of weird, but yeah.